A recent case of a young man needing bilateral lung transplantations after an infection caught my attention. And it caught my attention because I'm doing some work around preparing for my upcoming presentation on uh, COVID and an unusual trapdoor or backdoor that it uses to infect cells. And this case was published on the 30th of January 2026, and it was all over the news. So I want to understand a little bit more about the case to see whether or not it fit with what I was looking at. In case you're interested, I'm doing a presentation about COVID trapdoor or backdoor and how it depletes your immune system. And so this paper really fit with what I was looking at. The only problem is that I couldn't see the full paper because it's blocked, not blocked, it's paywalled, and I don't intend to pay for the full ability to read it. But if you wanted to, you certainly can. But I have got some information from it, which I was using to make a case, so to speak. So it's important to know I don't have the full story, but I'm going to explain to you why I'm talking about it in preparation for this upcoming webinar that I'm doing. Look in the description for the link. And in it, they had to replace the lungs of this gentleman. He had very severe acute respiratory distress syndrome and caused by a drug-resistant infection. So he had clearly some kind of bacterial pneumonia. But when I looked at the histology, there was something unusual about the histology. So this is what caught my attention. It's important to know that he was transplanted, and they were doing this paper because it's 2 years after he's been transplanted and he's still doing well. So that gives me a timeline of about 2024 or late 2023. It's right in the midst of the pandemic. And so what the likelihood is that he had potentially, I can only say potentially, a COVID infection with subsequent superinfection or bacterial superimposed infection, because this is exactly why I'm talking about this presentation, because they have found a way that COVID literally gets into specific immune cells and destroys them. And to explain the concept, I had to be working at this image here. I'm trying to get my head around how to put this across to you, but I'll show you three images so that you understand what I'm looking at. Here is a castle, and the castle is supposed to represent a cell. They are battling to keep the enemy outside. And this is the main gate that they're battling to protect. And this main gate would represent ACE2. But what happens is that there is a tunnel, backdoor or trapdoor, where the enemy can get inside the castle using this backdoor. And this would represent CD147. The thing that I'm talking about in relation to how the viral infection can spread to cells that don't normally get infected because they don't have ACE2. Just in case you don't understand what I'm talking about, simple principle, the spike protein is the key. It uses a specific kind of lock, like for instance, a Yale lock to open the door. That Yale lock would be ACE2. And that's the primary mode that the virus uses to get inside the cell or open the door to the house. What they've found is that there's another door. There are a few other doors, but one that they have found quite recently is that when the virus gets in through this window, it's like it has an extra key that can open the window. Once it opens the window, it's then able to open multiple other windows so that other viruses can, other viruses can get in. And so this is the image that I'm thinking of. I'm still working on this, so you have to forgive me. Again, this is supposed to represent a cell, and this is the castle in the cell. It's defending itself against all the viruses. And then there is this backdoor allowing it inside to infect the cell, okay? Now, what is unusual about the paper that I have mentioned is the fact that once it gets in the cell, because many cells would try and close the front door, which is ACE2, but once the virus gets in through one of these back doors, guess what happens? It then opens multiple trapdoors. And suddenly, in the context of the castle and the cell, you have more virus able to enter and then infect that cell, possibly replicate and infect other ones. This is extremely important because it explains one of the unusual patterns that we'd seen in the pandemic, which is that lymphocytes, a specific kind of immune cell, was depleted. And I'll be covering this in more detail on Thursday. If you want to join me in the presentation, as I said, remember to look in the description. That's where you will find the link and you can ask questions. We usually have a good time discussing this. So this is what it's about. Now, you may wonder then, what is the connection with this case that I had mentioned right at the start? This is a young man who had lung failure, needed both lungs replaced, and he had a transplant, and he did well after 2 years. Now, why did I think of COVID in the context of that case? So I didn't have all the paper, as I said, but when I looked at the detail here, when they looked at the findings, so after they took out the lungs, that's the pneumonectomy, vasopressor requirements resolved. And what that means is that we usually have to use specific drugs to increase the blood pressure because the person is so unwell. So that could just be because the sepsis was removed in the lungs, or maybe it had less pressure going through the machine that allowed the heart to pump more efficiently. So they kept him like that until a transplant came along. And just so that you understand the mechanism, this is it here. It's showing you the lungs that have been damaged. And what they did is that they got this machine, an extracorporeal machine, to do the oxygenation of blood. So they connected it to the right side of his heart. So he gets venous blood going into it. It then oxygenates it here and puts it back into the left side of the heart. And then he is able to pump it around the body. We had to use this in COVID quite a lot because for many patients who had severe microclotting, they couldn't get any oxygen in the lungs. And so you had to use machinery like this to try and see if you could keep them alive. This was able to be done for 48 hours. They kept him alive for 48 hours, enough time to get new lungs. They transplanted him, and he survived so far for 2 years. So this was what was done. And when we look at the pathology now, and this is why I'm saying we need so many autopsies. I keep saying the same thing. And it's like nobody's interested when this is probably the most important thing that we can look at. The characteristic with regards to these autopsies here, or this, in his case, it wasn't an autopsy. He was still alive, but they got the whole lungs. And when they looked at it, it revealed diffuse, uniform destruction across all regions of the lung, infiltrated by neutrophils. These are like the pawns in a chest cell, basic infection cells, macrophages, activated T cells. And it was therefore an inflammatory pattern that was there, along with expansion of myofibroblasts. These are cells that lay down collagen and make the lung very stiff. And so he had a near complete loss of the alveolar architecture of the lung. So his whole lungs were completely destroyed by inflammatory changes. They didn't say abscesses. They didn't say multiple little, they didn't say fungal. So inflammatory changes in the context of the pandemic, to me, that's going to point to COVID. And so it therefore highlights to us the kind of damage that can occur with infection. I don't know the details on this case, but it raises the point, and this is what I'm now looking at, is that Where we are now is a different stage of the pandemic. We no longer really see severe COVID-19, but anybody who is looking at mortality figures will realize that, still a lot of people are dying. It's not as if after the peak it suddenly dropped down. What is the mechanism? COVID is still circulating. I'm very interested in what are the outcomes in unusual presentation, especially in vaccinated cohorts who are unlikely to have the typical pattern that we would see. And so this is where it comes back to what I had been looking at with regards to CD147. This is how the virus is able to enter cells from the back door almost. And when they get inside the cell, I'll show you here what it would look like. This is a cell here that is infected. Normally, it gets infected by ACE2, gets inside, replicates. But what they're finding is that another set of cells can use CD147 to get inside. This is the back door. And so that means it can infect a lot more cells, especially immune cells, and then it kills them. This means that the immune patterns that we are likely to be seeing are very different from what was there pre-pandemic because the virus is still circulating. It is just depleting the immune system over and over again with reinfections. And so if we are not careful, we are going to get caught out by very unusual presentation of disease, which is already happening. It's just that it is not a tsunami of unusual presentation, so it can easily go under the radar. Still a lot of research to be done. Don't let anyone kid you that this is over. There is a lot more work to be done because we are in a situation where virus is circulating, still doing damage. We don't fully understand it. We have limited autopsies. Nobody can be sure. All we can do is pick the pieces together. And that's what I'm asking you to do. If you're interested, join, click on the description below. Let's see if we can look at the patterns. At that point, I'll be looking carefully at what diseases I would expect to appear. And it would largely be diseases with regards to limited or weakened immunity, fungal infections, reactivation of viruses, the same pattern that I think we are seeing already. It's just at the moment still under the radar. Thank you for joining me, and we will continue to explore the science in the next few weeks.